Welcome to this explainer. When we picture a cell, it's really easy to just imagine it as a tiny water balloon, kind of like a static shell just holding all the important stuff inside. But today, we're going to completely shatter that image. We're looking at the plasma membrane not as a simple, passive boundary, but as a bustling, microscopic city border. It's an incredibly dynamic, highly regulated, and surprisingly complex structure that literally keeps you alive. Let's get right into it.
Here is our roadmap for today. We'll be covering 5 key areas. 1. The fluid mosaic model. 2. Lipids, the fluid foundation. 3. Proteins, the membrane workforce. 4. Carbohydrates, the cell's ID. And 5. A dynamic, asymmetrical border.
But before we break down those components, I really want you to appreciate the sheer scale of what we're talking about here. 5. That number represents 5 nanometers. This entire bustling city border regulating absolutely everything that goes in and out of your cells is unbelievably thin. It ranges from just 5 to 10 nanometers thick. To put that into perspective, a single strand of human hair is roughly 80 to 100,000 nanometers wide. This minimal thickness is exactly what makes its complex functionality so absolutely mind-boggling.
All right, kicking things off with Section 1, the fluid mosaic model, a living ocean. To understand how something so intensely thin can actually work, we have to talk about the structural theory behind it. The fluid mosaic model was proposed back in 1972 by scientists S.J. Singer and Garth L. Nicholson. The absolutely crucial word to remember here is fluid. The membrane is not a solid brick wall. It's much more like a living ocean. It's a flowing mosaic where all the various components, the lipids, the proteins, the carbohydrates, they can actually change position and flow around each other while still keeping the fundamental integrity of the border perfectly intact. Molecules are rapidly diffusing across and side to side, creating this highly energetic, moving environment.
So, what exactly is floating around in this living ocean? Let's look at the actual ingredients. If we analyze the membrane's composition by dry weight, we see a really interesting split. We've got proteins making up 52%, lipids at 40%, and carbohydrates coming in at 8%. At first glance, you'd probably think, okay, the membrane is mostly protein. But dry weight doesn't exactly tell the whole story of what this border looks like at the molecular level.
And here is where it gets really fascinating. 50. Because proteins are incredibly large and heavy molecules compared to tiny lipids, the actual physical ratio is staggering. For every single bulky protein molecule embedded in that membrane, there are roughly 50 tiny lipid molecules surrounding it. That means the sheer physical volume and the actual foundation of the membrane are overwhelmingly made up of lipids.
Which leads us perfectly into Section 2, lipids, the fluid foundation.
Within this membrane, we essentially have 3 major categories of lipids keeping things together. First up, we have phospholipids. These are the absolute most abundant, and in animal cells, they typically feature a 3-carbon glycerol backbone. Next, we have glycolipids. These are super interesting because they literally define our blood types, like the A and B antigens on your red blood cells. And third, we have sterols, which is primarily cholesterol in animal cells. Cholesterol actually makes up about a quarter of membrane lipids and acts as a key thermostat, influencing just how fluid or rigid the membrane behaves.
The big question is, why do these lipids form a membrane in the first place? Well, it all comes down to a sort of structural split personality. All membrane lipids are amphiphilic. They have a hydrophilic or water-loving polar head and a hydrophobic or water-fearing non-polar tail, usually made of fatty acid chains. When you drop them into a watery environment, which, the inside and outside of a cell definitely are, they spontaneously assemble. The water-fearing tails tuck inside to hide from the water, while the water-loving heads face outward. This brilliant little quirk naturally creates the fundamental double-layered sheet of the cell, the lipid bilayer. Now that we've got our lipid foundation laid out, let's move to Section 3, proteins, the membrane workforce.
While the lipids are providing the structure, the proteins are the ones doing the heavy lifting. In fact, membrane proteins account for about 30% of all the proteins encoded by the animal genome. That's huge. Their essential roles are incredibly vast. They act as transport mechanisms, actively moving specific molecules safely across the border. They handle catalysis, driving vital chemical reactions, like synthesizing ATP for energy. They serve as structural links, physically anchoring the cell's internal skeleton to the outside world. And they function in reception, acting as these little cellular antennas that receive chemical signals from the environment around them.
But not all proteins interact with the membrane in the exact same way. We can generally group them into 2 major classes. Extrinsic or peripheral proteins are kind of like temporary visitors. They're located entirely outside the lipid bilayer, loosely attached by easily broken hydrogen bonds. On the flip side, intrinsic or integral proteins are permanent infrastructure. They are tightly bound, often crossing completely through the membrane. You would literally need harsh chemical detergents to forcefully detach them. They are completely locked in.
All right, let's move to section 4, carbohydrates, the cell's ID.
Carbohydrates make up the absolute smallest percentage of the membrane by weight, just about 2 to 10%. But they follow a very strict geographical rule in cellular biology. Carbohydrates are always, without exception, found exclusively on the exterior surface of the cell. They attach to proteins to form glycoproteins, or they attach to lipids to form glycolipids. Together they create this dense, sugary coating all around the cell known as the glycocalyx.
Now, this glycocalyx coat isn't just there for show. It has some incredibly crucial physiological roles. First is adhesion. It literally helps cells physically stick to one another so they can form tissues. Second is protection. It acts as a coat of armor, serving as a physical and chemical barrier against rogue enzymes that might try to digest the cell. And finally, recognition. These tiny carbohydrate chains, which can be anywhere from 2 to 60 units long, function as highly specific ID tags. They allow cells to recognize friends, foes, and neighbors circulating in the body.
This brings us to our final section, Section 5, a dynamic, asymmetrical border.
Remember, we are talking about a fluid mosaic. Things are in constant motion. Lipids actually exhibit 3 primary types of movement. First, lateral diffusion. They swap places with their neighbors at blinding speeds. A single lipid can swap places millions of times a second within its own layer. Second, in-place rotation. They just spin freely on their own axis. But the third movement, the flip-flop, where a lipid transitions to the opposite layer, is an entirely different beast. It requires energy and specialized proteins called fliposes to actually pull it off.
I want to pause and really emphasize this quote straight from the biological data. The transition from one layer to another is rarer, occurring less than once a week. Just think about that contrast for a second. Side-to-side movement is happening millions of times every single second, but crossing from the inner layer to the outer layer, that happens roughly once a week. This perfectly illustrates just how strictly the cell separates its inner environment from the outside world.
Because that flip-flop movement is so incredibly rare, it allows the membrane to maintain strict asymmetry. Essentially, the inside of the wall looks completely different from the outside. Lipid compositions vary drastically between the inner and outer layers. Proteins feature absolute asymmetry. Their outer-facing domains are built for recognition, while their inner domains are specifically built to attach to internal structures. And, as we just covered, carbohydrates are 100% located on the external side. It is a highly organized, dual-faced structure.
Finally, depending on what the cellular city actually needs to do, this basic asymmetrical membrane can adapt into some highly specialized structures. If a cell needs to optimize absorption, the membrane folds into microvilli. If it needs to stop leaks between cells, it forms watertight junctions. For sheer mechanical strength, it uses structures called desmosomes. And for instant rapid-fire communication, it forms gap junctions that allow chemical and electrical signals to flash directly from one cell to the next. The fundamental border structure easily adapts to become exactly what your body requires at any given location.
So let's step back and look at the big picture here. Everything you are, all of your trillions of cells, are defined and protected by a fluid, rapidly moving, highly intelligent barrier that is a mere 5 nanometers thick. It's an absolute microscopic miracle of structure and function. If an ecosystem this unbelievably complex exists on the incredibly tiny border of a single cell, what else is hiding in the microscopic world just waiting to be understood? Thank you so much for joining me for this explainer. I hope you walk away seeing the cells in your body in a completely new light. Keep learning, and I'll catch you next time.